It's a pleasure to introduce Brandon Bloomer, fifth year graduate student in my lab. So Brandon was born in Levittown, uh, which for people in the international community may not know that it's sort of the quintessential planned suburban American community. And it's basically six houses, all repeated and built in an assembly line. With workers moving from one house to the other. And in its heyday, a house was finished every 16 minutes, as I understand it. So you can go to Wikipedia, there's pages and pages. It's a famous place, but it's about 50,000 people outside of Philadelphia, which is a big city in the East. So Brandon isn't particularly conforming and is not one of six cookie cutter graduate students, although he does have an array of undergraduates that may be kind of an assembly line to try to maybe produce a protein every 16 seconds through directed evolution. So from there, he went to Temple University in Philadelphia, did undergraduate research with Sarah Wen-Granuk there on hypervalent iodine reagents, if I remember right, is what he worked on. That's her program. So not something in biochemistry or chemical biology or synthetic biology. But Brandon is very, very smart. And so he very quickly picked up on all the biological techniques and the chemistry behind the projects and has several things going. He was a major contributor on transport of artificial cofactors into the cytoplasm to bring our artificial metalloenzymes into that milieu. And is co-author on several papers that are submitted and have just been published, but also shares, I think, one of my loves, which is reaction mechanisms. And so he has been studying the mechanism of how some of the artificial metalloenzymes work, and that has led to actually realizing what some of the questions are. So one quick funny story on Brandon, when he was looking at graduate schools, as I understand it, he was in a library reading one of our papers on artificial metalloenzymes when we were making decisions on grad schools. And so I called him to tell him he was accepted while he was reading that paper. So it must have been a bit spooky for him, and then whether that made him come or, cemented the deal, I'm not sure. But anyway, we're very pleased that he's come to Berkeley. So take it away, Brandon.
Thanks so much, John. I don't think I was as spooked out as I don't know. It was a really interesting coincidence to have one of my chemistry heroes call me while I was reading one of his papers. But needless to say, like John said, I'm a fifth year graduate student in the Hartwig group, particularly in the biocatalysis subgroup, and I'm super excited to tell you about one of the many details that kind of unfolded in a mechanistic study that we've been conducting for a couple of years now on a carbene transfer catalyzed by an iridium-containing artificial metalloenzyme. So without further ado, in the Hartwig group, one of the strategies that we've employed to make organometallic catalysts is to take enzymes that naturally bind metal cofactors, such as this P450 enzyme that binds this natural iron cofactor. I'm sure people in the audience are well aware of these enzymes, but we've engineered them to bind this organometallic iridium methyl mesoporphyrin cofactor. And we first employed this strategy in 2016 to conduct these carbene transfer reactions that were at the time not known to nature. Subsequently, native P450 enzymes have been engineered by Arnold and Vassan to catalyze these reactions, which has certainly risen the bar on artificial metalloenzyme activity. But with that said, iridium P450s still catalyze pretty unique reactivities. For example, while iron P450 cofactors iron P450 enzymes have been engineered to cyclopropionate these activated alkenes and in some cases terminal olefins. The iridium manifold really stands out in its activity for these really challenging internal olefins and olefins that are appended to various terpene natural products. And so we're really interested in further studying these enzymes, especially in the context of their mechanism and the type of activity that is imparted by having the protein scaffold around a completely synthetic cofactor. And so this led us to a number of mechanistic questions. I think first and foremost, we're really just interested to know what the actual structure of this iridium P450 is. For example, we had evidence that the cofactor was in the active site because our directed evolution campaigns targeted the active site known from the original crystal structure of our P450. But I guess that still left us with the question, is the cofactor exactly where we think it is in the native heme binding pocket? And if so, what's the orientation? Because this iridium cofactor has a methyl group on it. So what's the direction of that methyl group? Where's that pointed with respect to substrate channel, for example? We're also really interested in conducting molecular dynamic studies. And so we, in collaboration with the Howe group, in particular Mark, who was a postdoc at the time we started this project, we're really interested in seeing exactly how the enzyme imparts diastereoselectivity. Because, for example, the cofactor is active without the enzyme surrounding it. However, it doesn't exhibit a very significant diastereoselectivity or enantioselectivity. So what's the origin of selectivity that is imparted by this secondary coordination sphere? And third, and probably one of the most difficult questions of this project were just probing the kinetics. What's the kinetic mechanism of the enzyme? For example, the classic dichotomy is between the ping pong mechanism, where the enzyme is first modified and then transfers a group to a substrate, or a sequential mechanism where two substrates have to bind together before any productive reactivity is attained. And we're interested in learning the rate determining step of the reaction as well. And on top of those classic kinetic questions, we also wanted to just know how the cofactor is interacting with the enzyme scaffold. Is it dissociating and reassociating? Is there some dynamic quality to the interaction between the cofactor and the enzyme? And so these are questions that were bugging us for years. And I finally developed conditions to probe the reactivity and the kinetics of the reaction. So for the sake of time, I'm going to be focusing on the structure of the enzyme, and secondly, the kinetics, in particular, this question of cofactor dynamics, which you'll see became a very relevant question after we first got our structure of the enzyme. So with Sean Natoli, who went up to the large Berkeley National Laboratory right up the hill from us. He screened around 20,000 different conditions with a robot to probe the conditions for the crystallization of this enzyme. And it's worth noting that this is actually the first structure of a P450 containing a completely synthetic cofactor. There have been structures of other globins like myoglobin or horseradish peroxidase that contain these artificial metalloporphyrins. But this is the first structure of P450 containing an artificial cofactor, which is really interesting in itself. So just a quick note about some of the mutations that we've made over our numerous studies with this enzyme scaffold. Most of them just change the active site. However, one of the really important mutations highlighted in pink here is between these F and G helices. And on this flexible FG loop region, we have this tryptophan residue, which at least in the solid state seems to pin back that loop, which opens up the substrate channel quite a bit. And we think that's responsible for the high level of reactivity for intermolecular reactions in particular. And so with that said, this was kind of an intuitive structure. We found that the cofactor is binding exactly where we thought it was in the active site, the native orientation of the heme cofactor. However, something might stand out to the viewers here is that the methyl group that's appended to our iridium methylporphyrin is pointed up into the substrate channel and active site, which is not the intuitive direction we would expect this group to be pointed for such an active enzyme. In fact, you can see just below it, there's really only enough room underneath the cofactor for that methyl group to be. So it's unlikely that the reaction's occurring beneath this porphyrin in the active site. And so this led us to, question our previous results. Is the majority of the enzyme inactive and it's just stuck upside down? And is that what we observed in the crystal structure? Or does this enzyme actually have to rearrange in a dynamic fashion to become the active enzyme that we know it as? And so one way we might expect the activation of this enzyme to manifest itself is in the form of an induction period. And so I took this model reaction, which is the cyclopropionation of carbone, with this diazo compound called EDA. And this process liberates nitrogen. In fact, that was a huge stumbling block for us because it liberates nitrogen so fast that it disrupts the path lengths of our UV-Vis and FTIR setups. It disrupts the field homogeneity in our NMR experiments. So we were actually picking our own brains as to how we would even analyze this reaction. And eventually we used our weakness as a strength, and we just measured the pressure of the nitrogen evolved from the reaction. And you can see these two traces here. This first trace is from the free cofactor, which has a low diastereoselectivity selectivity of 1 to 1 to 3 to 4. Being from the 4 diastereomers you can form from the cyclopropionation. And this red trace here is the enzyme catalyzed reaction. And this has a much higher diastereoselectivity selectivity of 8 to 1 to 1 to 1. So we know that the enzyme's assembled and it's imparting selectivity, but very interestingly, there is a very large induction period with this enzyme. And this induction period is only about 45 seconds long. It's important to know this reaction only occurs over the course of just a few minutes. But this induction period lends credence to this idea that the enzyme has to activate in some way that's unique to the enzyme itself and not this synthetic cofactor. And so while this lends credence to our idea, it still leaves the question, can a change in the cofactor's orientation actually account for this induction period that we're observing? And so this leads us to our mechanistic questions and hypotheses, because this difference in the induction period and the enzyme clearly indicates that there's some secondary coordination sphere effect being imparted by the enzyme. But whether or not that's a flip is still a question. So how do we even determine if a flip occurs during the induction period? And this is what the catalytic cycle would have to look like. You would have to start with some inactive form of the enzyme. It would have to undergo some transformation to become active, which I have suggestively labeled flip here. And then it would undergo the, I guess, stereotypical carbene transfer mechanism, which in this case is the formation of this carbenoid species that liberates nitrogen. And a detail that I haven't gotten into here is that we found that this is likely the rate determining step of the reaction, the formation of this carbene. And then this carbene then undergoes a quick transfer. And then this species, this active species, has to stay on cycle. And so the question is, can a flip actually account for this induction period? And so if that's the case, then this cofactor should be partially associated or dissociating from the enzyme scaffold and then coming back. into the active orientation. And whether or not that occurs can be determined by measuring the dissociation, the rate of the dissociation of this cofactor, and seeing if that maps onto the length of our induction period. For example, if the cofactor is flipping more slowly than the induction period, then we know it can't be a flip. But if it's flipping much more quickly than the induction period, then that can further lend credence to this idea. So we can estimate this, the rate of the induction, or the rate constant of the induction period, through simulating our experimental gas evolution data. And then we can experimentally determine, or rather we can determine indirectly, the rate constant for the cofactor dissociation indirectly by experimentally measuring the dissociation constant of the cofactor, as well as the rate constant for the binding of the cofactor. And so just to summarize that in a graphical way, this holoenzyme has to dissociate into the ABO protein and the free cofactor. And this happens more or less reversibly until the substrate is added. And so the way we would determine the rate constant for this dissociation is by measuring the KD of the cofactor, and then measuring the rate constant for the association, and then just using simple algebra to determine the rate constant for the dissociation. So with that said, that's what I did next. But first, as I mentioned before, we can simulate our experimental data to get a rough estimate of the rate constant for the induction period. And so in pink, you can see my experimental data. It's in triplicate. The error bars are represented by this pink area here. And when we simulate the data with Copasi, we can actually get, actually sort of an underestimate of what the rate constant for the induction period should be, which is this number here. And so what this will mean for us in the future is the rate constant for the dissociation of the cofactor has to be greater than or equal to this rate constant for the induction period. Otherwise, we can't conclude, or this does not support the idea that the dissociation of the cofactor can lead to the induction. And so with this number in hand, how does the K off compare to the K induction? And so, as I said before, we determine K off indirectly by measuring the KD and the K on values. To determine K or KD, we did a fluorometric titration of the ABO enzyme with an underequivalence of the cofactor. And by treating that data with a log-log plot, we can obtain a y-intercept that equals the minus log of the KD. And so that allowed us to determine the KD of 4.1 micromolar, which compares favorably to heme shuttle proteins, for example, which very weakly bind heme. And so that totally makes sense. And so we also determined K on, which is the rate constant of the cofactor binding. And we also did this through fluorimetry. And it's probably important to note here that when the cofactor binds, it actually quenches some of the intrinsic fluorescence of the protein. So we can do this statically through a titration method, or we can do it dynamically by measuring the time course of that quenching. And so we measured the time course of the quenching under saturating protein conditions. And what this told us after fitting with a single exponential decay was that we could determine that the K on was about 6.6 times 10 to the 4 per molar per second. Now this value is pretty fast, and it also compares favorably with known values. For example, the binding of heme to a protein called bovine serum albumin. And so with these values in hand, we can indirectly determine the K off, which is about 0.27 per second. Now, this is actually a very fast dissociation relative to native heme binding, which is what we'd expect because this cofactor is not anchored in this in the same way with an axial residue like heme is to native proteins. And the other important thing to note about this value is that it's much faster than or it gives a much faster dissociation of the cofactor than the induction period. In fact, this rate constant is about 20 times greater than the rate constant for the induction period. And so what that tells us is that a flip by a partial or full dissociation is it's very fast and can definitely account for the induction period. So with that said, we still wanted to do an independent measurement to assess whether or not the active site or the open coordination site of iridium was appearing on the time scale of this induction period. And so this ended up being quite challenging because the spectroscopic properties of the iridium porphyrin itself don't change very much in the presence of an exogenous ligand. So it was very hard initially to get a time course for any binding events. However, I developed a novel coordination probe to actually see how fast this open coordination site on iridium was appearing. And so this is based off of the Bo-Dippy fluorescent dye, and it's conjugated to imidazole, which can coordinate to the remaining open coordination site on our iridium porphyrin. And so very gratifyingly, this fluorescent dye, while it's yellow in the absence of iridium, emits green in the presence of the iridium hollow enzyme. And this is distinct from the APO enzyme, which barely changes. And so we see this very distinct change upon the binding of the Bo-Dippy dye to the iridium. And so that gives us the ability to observe the time course of this binding event and see if it actually corresponds to a flipping event within the hollow enzyme. And so if that's true, this cofactor or this dye should only be able to appear as fast as we know that the cofactor can flip. And so how does the ligand binding rate compare to the rate of the flip or the enzyme activation? And so at saturating concentrations of this Bo-Dippy dye, we measured the maximum rate that this dye can bind to the iridium porphyrin within the active site. And again, by using a first order decay, we were able to determine that the maximum binding rate constant for this cofactor or for this dye is about 0.34, which is astoundingly close to our value of 0.27 for the rate constant of the dissociation of the porphyrin. And so in this case, L can really only bind to the enzyme as fast as the cofactor can flip. And so this allowed us to come to the conclusion that cofactor flipping adequately explains the induction period caused by our enzyme. And this is super fascinating because that means our completely synthetic cofactor binds inside the enzyme upside down, and it's flipping until the substrate comes around and traps it in an active conformation. And so in summary, we've successfully identified the solid state structure of not only an iridium containing P450, but a P450 that contains a completely artificial cofactor. And so with a combination of gas evolution and spectroscopic techniques, we've also been able to demonstrate that the cofactor is bound dynamically and that it can bind ligands on the time scale of the induction period of our carbene transfer. And so in the end, we propose that the cofactors bound dynamically and that the induction period results from a flip of the cofactor occurring by reversible dissociation of the iridium porphyrin. And so with that said, I'd really like to thank the audience. I'd especially like to thank Allison, Anthony, and Anya for hosting me today. And in terms of the lab, I'd really like to thank John. This was a super challenging but exciting project, and we both spent hours and hours just thinking and talking about this. But I'd also like to thank our lab administrative assistant, Annika, for keeping the lab running while we're thinking on these very hard projects. And I'd like to thank our bio catalysis subgroup, which we refer to as BioBay. And I'd like to thank the Heartweek group for general advice and support. And again, I'd love to thank the audience for listening today. So thanks.
Okay, thanks very much, Brandon, for a fantastic talk. That was great. I'll hand over to Anya for the Q&A session.
Yeah, so thank you so much, Brandon. Great talk and great mechanistic study. We have a question online from Lucas South. Lucas, would you like to unmute yourself to ask the question, please?
Yeah, hi, Brandon. Great talk. Really like the mechanistic study that you did. I have a question about the cofactor incorporation. So do you know, well, can you actually measure free cofactor in the reaction versus a bound cofactor, and whether that affects your diester selectivity or, you know, any other mechanistic studies that you've done with the enzyme?
So that's a great question. We haven't determined the exact ratio of bound to unbound. We think that all of it is bound at the concentrations that we study the reaction, but when we decrease the concentration of the enzyme below the KD of the cofactor, we see an almost complete loss in the selectivity, which suggests that below a certain concentration, most of the cofactor will dissociate, and it follows a smooth curve that quickly descends. So there certainly is probably a very small amount of cofactor that can dissociate, but we haven't determined those exact ratios. With that said, at very high localized concentrations, for example, if we assemble the hollow enzyme inside E. coli, we see higher diastereoselectivity selectivity than we do in vitro. And again, I think that lends credence to the idea that at low concentrations, the cofactor can dissociate. So anyway, that's a great question. Thanks.
Great, thank you so much. So we have a lot of artificial enzyme experts. So Thomas, do you want to go next? If you'd like to unmute yourself.
Did you say Tom? Is that my turn? Okay,
yes.
So
yes.
Brandon, congratulations. It's really a very detailed, thorough, and spectacular study. And I'm sure you had many entertaining hours with John. So my naive question is, how stable is the iridium carbene? And could it be that you form the carbene outside, and that is what forces the proper orientation? I realize that, as you mentioned, that the formation of the carbene is the rate determining step. I was just wondering whether this could help to properly orient the cofactor, although the iridium carbene outside the protein also would catalyze the cyclopropionation reaction.
Yeah, so that's another fantastic question. We've actually compared the rate of nitrogen extrusion to the rate of association, and the rate of association is actually faster than the rate of nitrogen extrusion. So we think that the enzyme, once it forms the active hollow enzyme, it's trapped. It's trapped by the substrate, and the carbene forms rapidly enough that it likely stays contained within the enzyme scaffold. And again, that was done by comparing our K on value to our COPASI simulated data for the rate constant for the extrusion of nitrogen. So that's a great question too.
Okay, we have another question, this time from David Vargas from Rudy Fassan's lab. David, would you like to unmute yourself?
Hi, Anya. Yeah, thank you. This was a great talk. So I was curious to know if this is a phenomenon that you see in this metalloenzyme based on that iridium metal. If you change the metal to iron and manganese and other metals, would you also expect to see, the same dynamic formation that you show in this experiment?
Yeah, that's a really perceptive question that I've also asked myself. So I think it's possible that the cofactor can reversibly dissociate, but I think the iridium system is uniquely posed to be able to observe this flip, simply because it's just so fast. It's extremely active for forming the carbene, and so and subsequently transferring it. So I think that perhaps you would see it with the iron and manganese or even cobalt systems, but I think the extrusion of nitrogen will be so much slower than that dissociation that it would be a lot harder to observe. But if you wanted to exercise that or do an exercise where you could determine if it's flipping, you could probably just do the same type of determination of K off and then compare it to the evolution or the rate of evolution of nitrogen in your own system, or any other system for that fact. But in summary, I think that the evolution of nitrogen is just going to be too slow to observe a kinetically relevant dynamic system like you would see in the iridium.
Thank you.
If I'm allowed to jump in for 15 seconds, it's also because of the lack of axial ligand, right? So if you had another metal that also had the same absence of the direct binding to the protein, is probably the biggest factor.
Yeah, for sure. I was assuming that we have the axial.
Just to clarify that you're talking about the same thing.
Yeah, it's fine. Fine. Oh, sorry, Anya, it's fine to your question,
Yeah, so Mandisa, would you like to ask your question? If you could unmute yourself.
Yes, sorry. So congratulations on the nice and detailed study, firstly. I was just wondering a small thing. Is there enough space in the protein cavity to allow the flip? And why on the first space the iridium cofactor is binding in the upside down fashion, the methyl is on the substrate cavity?
So I just want to clarify the question. You're wondering if there's enough space in the active site that the cofactor could just flip inside it, and then the second question is, why does it bind upside down in the first place?
Yeah, that is my question actually, because there should be some interaction of the cofactor with the protein when it is binding to the protein, and if there is enough space for the or to overcome the how can I say the binding energy or the hydrophobic interaction that is already there in the cofactor and the protein to allow the flip.
Yeah, so those are both great questions. I think I'll answer the second one first because I think the answer is a little bit more straightforward. We think it binds upside down just because that methyl group provides some steric bulk. So especially when we're getting the crystal structure, what we're mostly going to see is that the cofactor is just going to bind with the path of least resistance, namely with the steric bulk up into the active site, as opposed to being smushed against the protein backbone. And then to answer the first question, that's something that we thought initially was that maybe it's flipping inside. I think the dynamics of heme binding would make that unlikely, because there's been a lot of studies for heme binding, and in almost all of them, there's a non-specific complex that's an intermediate in heme binding, and then that rapidly dissociates heme. And so what we're picturing is that there is some non-specific complex where our greasy porphyrin cofactor is bound non-specifically somewhere on the protein. We think it's unlikely that it's happening inside the active site just because it is a little bit more of a confined space. But the nature of the non-specific intermediate, there's probably many local minima that might look somewhat like it's still inside the active site. So I hope I answered your question. I think it's kind of a complex one, because since it's occurring on the time scale of seconds, it would be really hard to even simulate that. But our hypothesis currently is that there's just not enough space for it to undergo a fold flip inside the active site.
Yeah, thank you for answering.
Thank you.
Okay.
Well, thank you so much, Brendan. Back to Anthony.
Sorry, Brandon, I might use my privileges as host to sneak one more question in, if that's okay with everyone. Did you ever consider or think about it? I should say I'm not a protein NMR spectroscopist, but did you ever consider, like, for example, isotopically labeling your methyl group so you could directly probe the conformations in solution?
Yes. The first thing I tried and the hardest thing I tried was to get NMR spectra of this protein. And the problem is that the free cofactor aggregates in solution, which creates this huge molecular weight structure that tumbles so slowly that it broadens our signal to nothing. And then when we form the Metalloenzyme, that also tumbles too slowly. So we were just barely able to get a signal with a C13 labeled methyl group, but we were unable to perform any other subsequent, informative studies.
Okay.
So that's a great question. That's exactly what we tried first. And it was just a very challenging process.